Welcome viewers. Today the topic of our discussion is application of transposons in modern biology. I have divided the lecture into introduction, application of DNA transposon, where I will delve into details of different transposons in different organisms, for example the P elements, the transposons in C. elegans or the worm, the ACDS elements of maize, and the TN elements, which are the prokaryotic transposons, how it can be used in modern biology, and I will also discuss about certain reconstituted transposons which have revolutionized the use of transposon in modern biology. Further, we will discuss about the use of retrotransposon and also see how much is the possibility of application of transposon for therapeutic purpose. Now coming to the first part, the transposons or the so-called jumping genes were first discovered by Barbara McClintock, and what she found was that there are certain traits which are present in, she did it in Zea mays, and which were not stable and they were changing from one generation to another. And she suggested that there are certain parts of the genome which physically move from one locus or region to another loci. And this were later detected also in prokaryote and named as the jumping gene, or nowadays better known as the transposons. Now before I delve into the use of transposon, let me make you aware of the terms. Like I will use the term transposons, which is the transposable element or the DNA element. Then there are certain things known as the transposase, which is the enzyme which helps in cutting the transposon from its original site and helping it take it to another site. There are some inverted repeats which are present, which is identified by these transposase. And lastly, the whole process of this movement of the genetic material is known as transposition. Now coming to the use of these transposons, mostly, under broad category, they can be used to disrupt the activity of the gene because they can go and sit inside a gene. They can be used to clone a gene because they can sit on the two sides of the gene. They can also be used to identify gene and as well as carry a gene from one part of a genome to another part or one part of the genome to other organisms. Now, the transposon broadly categorized as DNA transposons and the retrotransposon. I will first talk of the DNA transposons. So the importance of DNA transposon is that they can be treated equivalent to naked DNA. And further, we can, by controlling the activity of the so-called transposase enzyme, we can control the movement of the transposons. We can actually control where to take these genetic elements and when to take these genetic elements, and further we can use these as vehicles for delivery of different genes or even for the disruption of different genes. Now these transposons has been widely reported in eukaryotes as well as certain prokaryotes and mostly found in the yeast, the C. elegans, the Drosophila, and also in plants like Arabidopsis and rice. Now first we'll discuss about the P element, which was one of the noted discoveries a few years back, and how it can be used as vehicles for gene disruption or gene delivery. Now first is we can use P element to cause insertional mutagenesis. So what is it? Now let's say this is the actual position of the P element. Now because the P element can hop, that is move around because of its transposase activity, it can go and get inserted at different locations. Now if the gene of interest is present in the vicinity of such locations where the P element will get inserted, then it will disrupt those. So this is what we know as insertional mutagenesis. Another important thing which take advantage of the insertional mutagenesis is transposon tagging. Now transposon tagging is a method by means of which we can actually tag a transposon, that is we can put the transposon behind or in front of the gene and the whole thing now acts as a cassette and we can identify such gene, we can clone this gene using the PCR because we know what are the ends of the transposon and we can use this cloned gene as a vector to move around. Now, if you know about various applications of this, the first important application of these events was to prepare the enhancer trap lines. So what are the enhancer trap lines? So I think we are aware of what are enhancers. So they are present before the promoters, or they enhance the activity of certain genes. So what people do is that because the transposon move around from one place to another, they can go and sit beside an enhancer. So, and they can also, while it moves around, they can take the enhancer from this region and go to another place. Now, whenever this cassette sits beside an enhancer or take this whole enhancer along with it to a new locus, so whatever gene which is behind this, or whose promoter is behind this enhancer line which sits into a new locus, will get transcribed. So we will see the expression. So as you can see in this figure, first there is a genomic enhancer. The P element sits behind the genomic enhancer. Now the P element, the people have used some reporter genes which are tagged to the P element. So whenever this P element is expressed by the presence of the enhancer, we will see the expression of the reporter gene. One of the very important use of such reporter is the LAGZ system, or even a GFP molecule. So under the fluorescence microscope it will look green, and it will also indicate that the gene after this is getting expressed. Hence, by using enhancer trap lines, we can spatio-temporally control the expression of different gene, and this has been used by scientists to know the function of different developmental genes in Drosophila. Another system is known as the enhancer detection. What people do here is they put this P element in front or beside a GAL4. So I think we are aware of the GAL4 US system. So the GAL4 are certain proteins or small molecules which will go and bind to certain regions which are known as the upstream activating sequences, and once they bind to the upstream activating sequence, they will help in the transcription of the genes which are behind it. So now if I have the P element sitting beside the GAL4, as the GAL4 move around from different places due to the hopping around of the P element, it can go and sit beside different gene and lead to the expression of those genes. Now these genes, one thing we should note, should have the upstream activator sequence in front of it to make this whole system work.
Sir, can P elements be used to deliver genes?
Yes, P elements can not only be used to move around certain things, but P elements can act as vehicle to transport gene, and this was first propounded by the famous experiment of Rubin and Spradling in 1982. So I will discuss the gist of the experiment. So what they did was they put a eye-colored gene which is called the rosy, which gives rise to red color of the eye, in a cassette of P element, but somehow they cleverly deleted the transposase, which is the enzyme of the P element. So because this enzyme is deleted, this P element by itself cannot move around. So they made a plasmid which has this P element and the rosy gene. They made another helper plasmid which has the P element along with the transposase. They injected both these constructed plasmid as well as the helper plasmid into the germline cells of Drosophila, but these Drosophila were homozygous mutant for the rosy. So they were not red-eyed, they were some white-eyed fly. Now, because the P element can move around, because the other helper plasmid is providing the transposase activity, now there is a chance that this will get integrated into the host genome. So in the first on the next filial generation, when we mate them together, so the progeny we get, there will be some flies where the eyes will be red, suggesting that we have used P elements as a vehicle to move the rosy gene, which was not present in that genotype, into the genotype.
Can P element vectors be used for gene expressions?
Yes, P element vectors can be used to control gene expression. How will we do that? So we have discussed about the GAL4 US system. So now if we have a GAL4 which is tagged to a P element. Now, because the P element can move around from one locus to another, so the GAL4 will similarly move around. Now, if we have some US tagged genes which are present at certain tissues, and if the P element along with the GAL4 goes and expresses in those tissues, then the GAL4 will go bind to the US activator sequence, which is the upstream activator sequence, and help in the expression of gene. Thus, the P element which is tagged to the GAL4 here acted as a vector which can control gene expression in different tissues, and this, to be correct, may not be the endogenous tissue where that gene is expressed. So what we use the term, we call it ectopic expression. That is, it can be used even in tissues where the gene is not supposed to get expressed. Now, coming to another important consequence of the use of P element is we can use P element to do replacement of genes. Now, how will we do that? So first thing is that whenever a P element go and bind to a particular locus, it leads to double-stranded cut. Now, so there is a gap. So the gap needs to be filled, and certain things are taken as a template by the host repair machinery to fill this gap. Now let's take, I want a gene A to be put in a region where gene B is present. So I make a P element vector where the sides are similar to the gene A. Now when this P element vector gets inserted at the region of gene B, the template of gene A will be used for gap filling. So at the end of the day is the gene A, which is now sitting in the locus where the gene B was present. This will lead to proper gene replacement. Now whether it's a total gene replacement or not depends on the design of the vector. Next we will come to an important limitation of the use of this P element. Now whatever I have talked till now is only valid for Drosophila, because these P elements and its transposase activity is non-functional in other organisms. So let us try to study the transposable element in other known organisms. One of the important one is the C. elegans or the so-called worm. Now in C. elegans, the TC based element, the TC1, the TC3 are present, and this, similar to the P element, can move from one part of a genome to another part, and thus can be used as good tools to do gene disruption as well as transposon tagging, that is, identifying unknown gene. However, while doing this forward genetic screen, one important thing is that these movement of the TC1 or the TC3 elements are quite rare. So what people have devised, they have used a MOS1 element in C. elegans, which is a member of TC1 mariner family, and the MOS1, the importance of the MOS1 is its movement is very specific and can be controlled. So we can look at this figure. So we can have some heat shock promoter which activate the MOS1. So whenever we provide heat shock, we know then the MOS1 will be activated, otherwise the MOS1 will not be activated. So then the MOS1 moves and it gets inserted at particular locus of the host transposon and help the host transposon to get inserted at certain locus of the genes and causing gene disruption. Now there are various mechanisms, PCR based, phenotypic, or even a genomic based method by means of which we can identify which genes has been disrupted due to the movement of the MOS1. The next important application of these transposable elements in the C. elegans was to do site-directed mutagenesis, and it was in 1992 when Plaster and his colleagues found that the TC1 elements can go and sit in the UNC22 gene, which is a gene present in C. elegans, and its movement caused double-stranded break, and thus what it did was it actually caused mutation of the UNC22 gene. Now people have also devised certain transposons which are tagged to GFP, which can go and sit inside a gene in a specific locus of the gene, disrupting the gene per se. And the expression of the GFP itself tell that the gene has been disrupted.
Sir, usually transposons shuttle within the same organism. So are there evidences that they can be used between different organisms as well?
Yes. So till now what we have discussed, C. elegans as well as Drosophila, the transposons are present within them and they are restricted to them. But if we come to the ACDS elements of maize, they are not just restricted to maize. We can use the transposable activity of the ACs and also its activity to move around the DS elements in rice, in barley, in wheat, and it has been widely used to do gene inactivation, gene tagging, transport of different gene from one locus to another, and has been done. Furthermore, I should make it clear to you that there are the AC elements or the DS elements which are called activation tagging. What people do is they encode the DS or the AC elements in front of the gene in such a way that whenever they move around from one locus to another, they move along with the promoter enhancer region of the gene. So whenever they go to a new locus, they can actually help in the expression of those genes. Okay, so irrespective of the endogenous promoter which is present. The third important category is the presence of the prokaryotic transposon, for example the TN elements, the mini mu elements, and what they can do is they can also do site directed mutagenesis. They can also be used for application of movement of different genes from one locus to another in prokaryotes, or they can act as vector for shuttling of different gene or incorporation of different gene in prokaryotes. Another important aspect of this is the signature tag mutagenesis. So what is signature tag mutagenesis? As you look at the figure, there are certain DNA signature tags, and these DNA signature tags are incorporated on the transposons or on the transgenes. So these are transposons which are present and these are sitting on the transgene. Because it is sitting on the transgene, it will affect the expression of the genes present in this transgene. Now, if let's say I want to screen for certain genes which are giving some cancer or some viral infection, so if we do such signature tag mutagenesis, so the genes or the virulent gene may be disrupted because of that. And if I now do microarray of the untagged versus the tagged mutagenesis, we will have a clear idea what are the virulent or what are the genes which is resulting in virulence, because those genes might be turned off while we do signature tagged mutagenesis. Now coming to another important aspect is the use of reconstituted transposons. Now the importance of reconstituted transposons is they are derived from certain inactive transposon system. However, the basis has been so changed, now they are active transposons. For example, the SB elements, which are better known as sleeping beauty elements, so they are inactive, that's why the name sleeping beauty. They can be tweaked around so that now they can move from one place to another. They can be given some transposable activity trans, that is there are some transposon present somewhere which provides the transposes because of which they can move around, or they can encode their own transposers. And what they do is they can do high efficient transposition because their original copies are missing in the host organisms. These transpositions are very specific in nature. The second such group is the Dada group family of transposons. The name Dada is known because they have sequence similarity to Daphnia as well as Dania renum, which is the frog. And these cause site-specific integration of the transposable element, due to which we can have gene disruptions in a very specific way. The third important element, which are the reconstituted, is the piggyback element. The piggyback were isolated from moths like Trichoplusia, and the importance of the piggyback element, unlike the SB or the Dada, they can create precise excision. So when they move from one locus to another, they don't take other things. They precisely excite itself and go to another locus, so the gene disruptions are perfect. However, I should also tell you that while SB and DADAs are not affected, their activity is not affected by the methylation of the DNA, but methylation of DNA leads to inactivation of the piggyback transposons. This is a cartoon which shows what are the uses of these reconstituted transposon. They can be used for gene transfer, they can be used for insertional mutagenesis.
Sir, we have heard of retrotransposons in eukaryotes. Can this be used in gene mapping?
Yes, very good question. Retrotransposon are indeed used in different eukaryotes for mapping of different genomes. I will come to it just now. So, a very good example is the grass family. And in grass family, there are different retrotransposons, and most important thing is the different copy numbers of the retrotransposons. And because of the difference in copy number of the retrotransposon, though they all, let's say rice, let's say the sorghum, the maize or the barley, all are members of the grass family, but the genome sizes vary. And in this figure, you will see that the green part actually represents the presence of transposons, and the orange part, the active genes. You will see the active gene for a particular chromosome may be less in barley and more in sorghum or rice, but their genome sizes differ because the amount of retrotransposon present in different. Now, so there are enough amount of area present in certain members of this grass family where the retrotransposons can move around still without affecting the function of the different genes, and hence retrotransposon based marker system has been widely used to reveal the polymorphic nature and the sequence specific amplifications which are present in various plants and various other organisms, and these are known as the sequence specific amplification polymorphism, or SSAP. And the sequence specific amplification polymorphism provides better estimate for studying the phylogenetic relationship in plants, and these are much better than RFLP or AFLP methods. The next important thing is the use of certain transposons like TN1 elements, which is found in tobacco plant, and these can be treated as vectors to transport different genes into the plant. Even the TNT1 can be changed with certain specific markers, for example the antibiotic resistant markers, so that when they go and sit in a particular locus, they not only provide disruption of a gene or take a different gene, but we know from outside, because the marker is sitting, so we are very sure that that transposon has gone and sit in that particular locus. Okay, another important use of certain transposons in plants is that they can alter the gene expression, and they can go and sit at the promoter region, they can go and sit at a particular locus within the ORF, and they can inhibit expression of different genes. In humans also, retrotransposons are very important. If you have heard, humans have the signs and the lines, that is the short interspersed nuclear repeat and the long interspersed nuclear repeats, and the presence of the signs and lines are very important, because let's say the signs and line composition of you is quite different from me. So in DNA fingerprinting, we can use particular sequences of sign, and we can digest and chop up the DNA. Depending on how they are arranged, my DNA will be having a different signature from your DNA. And hence, we see that retrotransposons are so very important in genomic studies, not only in plants, but also in humans and other organisms. Now we come to the therapeutic application of the transposons, and we have seen that there are cassettes of transposon which can catch the oncogenes depending on sequence similarity of the oncogenes, and these are known as transposon based screen for cancer genes, and is being utilized in practice, though it has not come as a wide scale level, but at the laboratory level it has been utilized to identify different cancer genes, and the transposon which is used is the reconstituted transposon, like the Sleeping Beauty. Further, transposon can be used as effective vehicles for gene therapy. Now, there are few important points. First, they are natural sources. Second, they are DNA, and they are naked DNA which can be inserted. Then, in most cases, especially for the reconstituted transposon, they are specific at which site they will go and bind. So, this is also on trial. What we want to do, what therapy we want to give. Let's say I want to take this gene which is inactive in a patient and make it sit there so the proper gene is being expressed, then we can use transposons as vehicles for the delivery of these genes. And this is a research area which has opened up, and maybe in few years time we'll see its application even in hospitals.